Bioinformatics is a major source of tool which helps the modern biologist to come to a quick and cost-effective preliminary hypothesis. The vast array of tools offered by bioinformatics forms the central contribution of the discipline. Let's get a quick taste of some of these popular tools. Let's move into a laboratory.
BLAST is a very important software tool that bioinformatics has contributed to new biology. It is almost like the Google of biology. Today, any modern biologist who deals with DNA, RNA and amino acid sequences first approach BLAST to verify what the sequence in their hand is. Let us see an example of an amino acid sequence which represents a protein called keratin, which is well known to be the structural protein in our hair, nails and skin. We see on the screen the sequence for keratin protein. To understand what keratin is and what it does and how other living beings, this particular keratin is from human, how keratin in other living beings are related to this, we can use BLAST. BLAST is available on the internet as a separate website, and we can approach this website to study about the keratin protein that we have. Let us now see how our keratin protein can be analyzed using BLAST. Ranganayagi will show us how the steps are taken.
Go to the BLAST homepage. In the given text areas, insert the keratin sequence you need to BLAST. You can copy, paste or browse the sequence from your desktop in FASTA format. Click the button BLAST to run the program. The result page will take few seconds to get loaded. Once the page is loaded, the results will be presented as 3 sections such as graphical display, a descriptive table and an alignment section. The graphical display of BLAST result will be displayed on the top of the result page. Level of similarities are represented using colored bars. Within the bordered graph, the top segment displays the color key and the query base scale. The position of each bar indicates the region of the query the high scoring pair covers. The next table gives additional details such as e-value, query coverage, maximum score, maximum identity and link out icons. The actual alignment is displayed here. The alignment header has the following fields such as score, expect, identities, etc. Here we could see the alignment of query protein with homologous protein in the database. The first hit will be the same as of query protein. Next to Homo sapiens, we got the hit from Calothrix jacus, common marmoset, a small monkey with an identity of 95%. Next hit is obtained from Pan troglodytes, a chimpanzee which is the closest ancestor to human with an identity of 98%. Following chimpanzee, Nomachus leucogenus, a north white-cheeked gibbon, got an identity of 97%.
While BLAST compares 2 sequences at a time, it is very often required that we compare a set of sequences together. Such a comparison is known as multiple sequence alignment. This is a computationally complex problem for which some practical feasible solutions are available through bioinformatics. Clustal is a tool used widely for multiple sequence alignment. We will now see how the protein keratin, which we saw in demonstrating BLAST also, being used in Clustal. We select 3 types of keratins, 1 from the human and 2 from the family of chimpanzees. We collect these sequences from databases on the internet and then give it to the Clustal server, which will show us an alignment of all of them. From this alignment we can see that these sequences are almost 95% similar across different organisms, and also we can see where they are exactly the same and where changes which are now known to have happened due to mutation are taking place. Let us see Ranganayagi's demonstration of cluster.
In this example, we had chosen keratin protein from 4 different organisms such as Homo sapiens, Calothrix jacus, Pan troglodytes and Nomascus leucogenus. The different output formats such as Philip, GCG, MSF, Pearson or FASTA are included. Click the submit button to run the program. It just takes a very few seconds to load the results. The result page contains alignments, result summary, guide tree and submission details. The alignments can be colored using the option Show Color. This option colors the residue according to their physiochemical properties. Red color indicates the small, hydrophobic amino acids. Blue indicates acidic and magenta indicates basic amino acids. Also, these alignments can be downloaded as a separate file using the option Download Alignment File for further use.
One of the very established activities of a traditional biologist is to categorize and name all forms of living beings that she comes across. A computational method for very objectively comparing organisms is today based on their DNA or amino acid sequences. Once a set of sequences are given, they can be aligned in the method which we have already seen called multiple sequence alignment, and then based on the alignment and some measurements which are made on the alignment, we can create a tree of the given organisms. Many biological observations are done based on such trees, including deciding drugs for new emerging viruses. Let us see how, given a set of sequences, a phylogenetic tree can be drawn using a software called MEGA. We will now demonstrate how, given a set of amino acid sequences from a choice of organisms, we can produce a phylogenetic tree. We will select a human being, a chimpanzee, a monkey, a donkey and a fish. Vidya will now show us how the software MEGA is used to produce such a phylogenetic tree.
Open MEGA tool by double-clicking on the MEGA tool icon. From the main MEGA launch bar, select Phylogeny, Construct or Test Neighbor Joining Tree menu option. A select window will appear on the screen. Now, you need to load the alignment file, locate your alignment file and click Open. Now, select a genetic code from the list and then click OK. Once you have selected, click Compute button. Now, you shall see the neighbor joining tree in the Tree Explorer window.
While most of bioinformatics is centered around DNA, RNA and amino acid sequences, which are, as we have seen, captured as simple text files which can be opened in software such as Notepad. However, there also exists an area where the data that is captured on a computer is an image, not a text data. These arise from a modern biology experiment known as microarrays, where the expression of a large number of genes in an organism are captured together in an experiment, and this experiment produces a digital image, and by using softwares that exploit the principles of digital image processing, we can derive conclusions and we can make measurements about which genes are active in a particular situation, which genes are actually active along with or in parallel with some other genes, which genes seem to be switched on while some other genes are switched off. All of this can be analyzed using bioinformatics softwares. We will now take a look at some of the microarray images and its analysis data. Ashwati will show us some of these images.
Open Magic Tool main window. From the project menu, choose New Project. Locate the folder in which you want to save your project. Type a name for the project. Magic Tool will automatically create a folder with this name and a project file in the directory. Gridding and clustering can effectively be done using Magic.
Biologists who wish to study structures of molecules very often had to rely on physical models for each of the molecule of their interest. Today, bioinformatics tools such as RASMOL and PyMOL offer a very attractive alternative to physical models. The structure of molecules are today recorded as data files, and this can be viewed conveniently in three-dimensional graphics in softwares such as PyMOL. We can, by a click of a mouse, change the type of the models. We can make measurements of distances between atoms or angle between a given set of atoms, and make such modifications that the biologist might require for the study of the molecular structure. These free softwares are very easy to use and very popular in study of biological molecules, and we will see one such software, PyMOL, which Rajesh will explain to us.
First, open PyMOL viewer from the Start menu. Then from the File menu, select Open. A selection window will appear on the screen. Locate the protein and open the file. From the object list, top right of window, you can select individual display modes. This can be selected using the S for Show, H for Hide, L for Label and C for Color buttons. From the click S button to choose Cartoon Display option. Then click H button to hide lines in the protein structure. From the wizard window, top of medium window, select measurement option to find the distance and then click 2 points on the protein molecule to find the distance between those 2 points. Then from the object list, top right of big window, click S for Show and select Surface Display option. Select Ligand, click S button for Show and select Surface option to display the ligand. To change the color of the ligand, click C for color change from the object list and then click red.
Computers don't speak human language, nor do they understand human language, and hence it is required for us to give computers instructions to cater to their specific need. For bioinformatics, specialized programming languages happens to be two languages, one which is called Perl and the other called Python. In our certificate program, we teach the programming language Perl. It is not a new language, and the speciality of the language is that it can process text very efficiently. As you must have observed, bioinformatics is centered around processing DNA, RNA and amino acid sequences, all of which can be expressed as text. We will see a very small Perl program which analyzes a DNA sequence and gives us a count of what kind of molecules are contained in this particular sequence. Umesh will show us this small program in Perl language, how it is written and how it is run and how the result is obtained.
The Perl program is for extracting the DNA sequence from the FASTA file. Here, example.fasta file is used as a reference FASTA file from which DNA sequence is extracted. Example.pl file is generated to perform the operation.
Skylab is an open-source scientific software package for numerical computations, providing a powerful open computing environment for engineering and scientific applications. Skylab can be used to plot any function or values in graphical form. Here you can see that a set of values is plotted as a sine waving using Skylab. Apart from values, we may also use Skylab for image processing. Here we use an inbuilt module in Skylab to open and display the image of an actress in Skylab. Now, the image can be further processed using other inbuilt functions available in Skylab.
Well, by now, we hope that you're ready to start your journey into bioinformatics with a clear bird's eye view. Wish you a successful program.